First of all, I'd like to thank Bionano for inviting me to come and speak and making me the last speaker, which is always just a really good time. I do have to say that, and I'll be very quick because I know we're running a little late, but last night I was very depressed. I don't know if anybody's a baseball fan, but the Yankees lost to the Houston Astros, so I was actually in my hotel right here when I was depressed, and then I saw these talks all day, and it made me very happy. So again, I'm going to talk about clinically relevant repeats in the human genome, a little bit of background on repeats in clinical medicine, focusing on the SCAs, the spinocerebellar ataxias. Then we're going to move into optical mapping, which actually should be called whole genome imaging, as I've been told. Focused on FSHD, DMPK, and CNBP repeats, a little summary and a little bit of the future, what we want to do with this technology. So repeat expansions, again, I'll try to be very quick. 1991 was when the first two repeat expansions were discovered, FMR1, of course, and SBMA in the androgen repeat. And they were both polyamino acid tracks, polyglutamine expansions. Relatively, I don't want to say small, but they were a little bit smaller than some of the stuff you can detect with the Bionano system. And altogether, there's about 22 inherited disorders that involve expanded repeats and also contracted repeats. And repeat expansion diseases include some of the most common inherited diseases, such as Huntington's and myotonic dystrophy, both 1 and 2. So my first week in the lab, I started at GeneDx about 2 years ago in July, and my boss came to me and said, okay, we want you to develop an ataxia panel. And my first question was, what is an ataxia panel? And I eventually figured that out. My training was mainly in research and a little bit of clinical medicine, but GeneDx was my first place to really be thrown into the clinical world. So an ataxia panel, they wanted basically all these different SCAs, DARPLA, Friedreich's ataxia, and SCA8. As you can see, these are spaced throughout the, this is an image of a gene. Friedreich's ataxia is intronic, SCA12 is in the 5' UTR, SCA8 is in the 3' UTR, and then a lot of these polyglutamine repeats. Some of what I already talked about. So SCA disorders, movement disorders, affect the cerebellum, brainstem, and also the spinal cord. It's basically a lack of muscle coordination, affects the person's coordination, their speech, balance, and it results from neuronal damage. There's also a lot of single nucleotide variants and some other smaller variants that can occur, 30 different genes. Most of the ataxias are autosomal dominant in inheritance. However, Friedreich's ataxia, which is a great one to look at on this system, is autosomally recessively inherited, and there's about 3 in 100,000 for the global prevalence. So this is basically a list of those ataxias. Some things I'd like to point out are SCA7 and 8, and Friedreich's ataxia can be huge expansions. Over 200 repeats. You cannot detect that on a capillary electrophoresis system. You can see a pileup, which I'll show you guys. But we knew these were going to be some difficult assays to actually get a good number for how many repeats were occurring. Some of the other things I'd like to point out is that these repeats are not just simple trinucleotide repeats. There's interruptions, there's other repeats. I know it might be difficult to see, but there's other repeats that can be expanded that are not pathogenic, but still can affect your sizing. So with all these problems in mind, we decided to dive in and try to design some triplet-primed PCR methodologies, TPP. Really, this is a very simple design, although I make it sound simple, but the people in my lab would argue it's not that simple. You simply have, again, I said simply, I apologize, 2 primers outside of your repeat sequence. You also have a repeat primer, which is usually 6 to 9 base pairs of the actual repeat. And what you hopefully get is this primer lays down, expands. This primer lays down, extends. Sorry, this primer lays down, extends, and you get multiple different products. And you basically get kind of this sawtooth of analysis. This is actually a Friedreich's ataxia carrier. And this is, I think it has roughly 500 repeats, which we haven't analyzed this yet with the Sapphire system, but we would like to. But you get a pileup. So all you can basically say about this is it's a carrier, and there is one repeat that's greater than 200 repeats, but we don't know the size. And this is, again, coming back to this whole idea of there's interruptions. There's a lot of difficulty in designing these assays. This is ATXN1, DARPLA, and there's actually a little insertion of three different sequences. We can actually design a repeat primer to go over those. So when we do the analysis, we actually get a little space here. This is one of the smaller repeats, so I don't know how useful a Sapphire would be for that, but just shows some of the difficulty with these repeats. And so with all those repeats that we want to do, I think it was about roughly 12 or 10, we decided we would look at doing some multiplexing of these repeats. So we use different fluorescent tags, FAM, NED, and also hex. However, we did get, we were able to combine these, have the same PCR conditions and everything like that. However, when we split these out, we do see bleed through of the different dyes. So with all that work, one other thing that makes doing a TPP assay difficult is you have to establish a correction factor. They also call it a MoCo, which is a mobility correction factor. So you can actually tell how many repeats there are. To do this, you have to find some homozygote samples. You Sanger sequence those homozygote samples. As you can see here, this is actually NA12878 genome in a bottle. And then you can have basically make a curve of different size homozygote repeats, and then you can figure out what your factor is to correct for the electrophoretic mobility. So this adds a lot of time to doing this type of analysis. Again, this is the clinical directors want to be able to say exactly or give a very close representation of how many repeats there are for these particular samples. So we actually were able to develop a panel of all these repeats. They also actually want to analyze all these repeats in a sample because of the closeness of the phenotypes of the SCAs. However, like I mentioned, there's some that we are not able to actually give an idea of how many repeats there are. And so it was suggested that we potentially look at doing southern analysis or other types of analysis, and that's something that I'll talk about a little bit later. So just a little bit about the design and implementation of TPP assays, in case anyone is thinking of doing it. We have 2 employees, took about 1.5 years to develop these 10 assays, actually 11. It's Lisa and Shamila, which I'd like to thank for doing all this work. That's 1.5 years of them just working on repeats, nothing else. And if you want to see people get really angry, make people work on repeats for 1.5 years. There was times when they really didn't talk to me directly, a lot of emails. The primer design is difficult. There's a lot of variance that can exist. This can cause allele dropout, poor performing. It's also reams and reams of data of optimization of the PCR conditions for this. So these 2 scientists had really great methodologies for going through and designing an optimal assay. Some of the Excel sheets they sent me had over 300 or 400 different lines. So it adds to the difficulty of it. And then also, and I mentioned this before, but you can't determine the size of the expansions for SCA 7, 8, and Friedreich's ataxia. This is due to the linear size range issue with capillary electrophoresis. And again, thanking those 2 people who did all this work. So then after we finished all that, my boss came and said, oh, so now we'd like you to work on myotonic dystrophy type 1, type 2, and also develop an assay for FSHD. And when I looked into these repeats, DMPK is in a 3' UTR. It's a 3 base pair repeat. You can get expansions up to 4,000 repeats. Same thing with CNBP. You can actually get up to, I think, 10,000 repeats or 5,000 repeats. And you guys have heard enough about FSHD today. I don't need to talk about that, the difficulty in that. So I thought, well, maybe I should look for another job, but I decided I would give it a try. So just to talk about myotonic dystrophies type 1 and type 2, as I mentioned, it's a CTG repeat in the 3' UTR of DMPK gene. It can get very large. DM2 is caused by a 4 base pair repeat in intron 1. That again can get very large. The key features are myotonia, cataracts, and one thing with being able to nail down the sizes of these repeats is that particularly DM1, myotonic dystrophy type 1, shows anticipation, where the offspring will have much, much larger repeats. So being able to identify, and that makes it more severe, makes it happen when they're younger. So being able to size these repeats is actually quite important. I won't go into FSHD. I think everybody's heard enough about FSHD today. So as I mentioned, southern blot was one of the techniques that was discussed. My company actually sends out a lot of these samples when they run a TPP assay for DMPK and CNBP. When they see only one allele, they send it out for a southern blot. So they said, oh, why don't we bring southern blot in-house? And I thought, oh, okay, well, we can give that a shot. However, when I was looking at what gels look like, and I know there's newer methods that are fluorescent tagged, but I still was a little wary of this methodology. It's also, no one in the lab had done a southern blot. I don't know if there's people here that have done southern blots, but I've heard they're quite tedious, difficult to do. So we decided to look at a different technology. And this was actually the time that I talked to Jeff, who was working for a new company and mentioned it was optical mapping. I had read a little bit about FSHD being possibly analyzed with it. And so I thought this would be a great thing to explore. So we really wanted to develop a method for calling these difficult repeats to size or difficult to PCR. And FSHD is probably one of the big ones that's really difficult to PCR. The other ones, you can amplify them, but as the repeats expand, you're going to have a lot of problems with it. And one of the other things we were interested in is developing a method to look at balanced translocations and potentially large clinically relevant CNVs, or structural variants. Balanced translocations, we do have a cytogenetics lab, and they do a lot of balanced translocations. A lot of samples that would come to GeneDx, they find the variant, it's unbalanced, they want to check the parents and see if it's balanced, and so they can make reproductive decisions. So this is really important. And just to give credit, all of the credit for this talk on the Bionano was done by Juan, Purnima, and Sandra. And sorry, the picture's pretty small, but they were instrumental in getting the system to work and getting the data processing going. So just some summaries. For FSHD testing, we've genotyped 40 samples for the FSHD contraction. These are all positive samples from Coriell. We also used, and I said internal samples, but we actually went through and found Coriell samples that had been used in papers that had negative FSHD results. I was very happy to find out the specificity and sensitivity were 100%, as was the PPV. So we were very excited about those results for FSHD. I'll skip through this. This is what a positive sample. You guys have seen enough images today of Bionano results, so I won't go through that, but a negative sample looks like. For DMPK, we've actually added to this since I made this talk. We've actually genotyped 10 samples that we knew had the expansion. Some of them are samples from GeneDx where we're able to get fresh blood and positive samples from Coriell. We actually had about 80% specificity on this. It should be a little bit higher because we've run more samples. But the positive predictive value was 100%. Just to show you what a positive expansion looks like, this individual is supposed to have 500 repeats, or around 500 repeats. That's what we were able to count. Our analysis, again, was it's supposed to be greater than 200. We don't know how many repeats there are. And then I'd like to talk about the sample that we actually weren't able to confirm. And one thing, well, I'll talk about it in a little bit, but it's supposed to have roughly 700 repeats. We don't see that. We then went back and did TPP on the sample. This is the full view. This is the pilot peak. So we did get a pilot peak, so we know it should have greater than 200 samples. So what we did was reorder the cell line. You can never be sure that either maybe Coriell, I don't want to blame Coriell, but maybe they sent the wrong sample. Maybe we switched the sample internally. Also to say that TPP was conducted on a genomic DNA sample, not the same sample we did the Sapphire on. So we'd like to explore that. So when we get that cell line back in, we'll extract the DNA from the cell line and both do the TPP and the Sapphire and compare that. So hopefully that'll clear up the problem with that. And CNBP, in summary, just to make it easy, specificity, sensitivity, PPV were 100%. We've actually upped this to 10 samples too, so I feel pretty confident that we'll be able to use this for larger expansions in CNBP. And this is kind of what the data looks like. You can see the normal allele, and I know you guys have seen enough of that. We also confirm this with our TPP. One thing that I'd like to point out with, and this is actually the R&D TTP, the one that's actually in production now, suffers from a lot of problems. Ours, you can see the expansion, but you don't get any pileup, which I still, basically if you cut it off here, it's expanded. Again, you don't know the number, but not having a pileup peak makes our clinical directors, makes our analysts very, very nervous. So they do not like not having a pileup. So we're still working on this one a little bit. And then, as has been mentioned in many of the talks today, we're actually using this for balanced translocations. This is a nice X to 17, and it was the only translocation that we saw in the sample, so we were very happy about this. So just to wrap it up, we've seen from a lot of our testing for the FSHD, DMPK, and CNBP, we started out doing cell lines, and then we started to get some blood samples in where we could get those blood samples before they went to clinical testing, just take 600 microliters. We were able to do that. The blood samples in our hands work much better than the cell lines. This was actually put in by my team. They said you must adhere strictly to the Bionano recommendations. I'm sure that's, you always want to pay attention to what you're supposed to do and follow the guidelines from the company. We've been using the manual workflow so far. I know that this workflow is automatable. We started working on it in our lab, but then I know that I think Bionano is working on automating it too. That would be very helpful. It's from watching the procedure and from talking to the people in the lab, it's a little bit of work. So the results are accurate, easy to interpret when it comes to insertions, deletions, translocations. And this is another thing directly from my team. They said the Bionano support has been extremely helpful with problems. I've been on some of the calls with the Bionano support, and it has been really fantastic and very, very helpful. So what's the future, at least in our lab, what we want to work on? Like I mentioned, we bank all the DMPK and CNBP samples that come in for testing. We actually might expand this to other repeats that we look at GeneDx. C9orf72 is another sample that we do a lot of those samples a week. We're going to start banking those, hoping that we can use that in a workflow and a validation for this technology in our production lab. We're also going to start looking at expansions in Friedreich's ataxia, SCA7 and SCA8. Although I believe SCA8, we don't actually have any positive samples, so we're still trying to get a hold of some of those. There's other additional repeats, such as SCA36 that would be interesting to look at. We intend to use the Sapphire for balance translocations. As I showed that one data slide, the data that we've looked at so far works really well for that. Again, we've had to work out with our cytolab being able to grab a sample when it comes in so that we can, once they get results, we can run our Sapphire system, make sure that they agree. And then as some of the data that was shown today, we'd like to explore this for methylation analysis, not only for FSHD, but also BWS and some of the other methylation disorders. Really like to thank the people at GeneDx who've done all this work. Amanda Lindy is a clinical director who works a lot with us on the repeats and tries to help us out for whether or not this would be something that the production lab would be able to do and whether or not they'd be able to analyze it. Then the repeats team, again, they worked, and they're still working on some repeats. And then also the Bionano staff, who was very, very helpful. And with that, there's my research-only use comment, and I'm open to any questions. Thank you so much for listening.